Alright, so we're going to be talking about the GWAS Catalog, which is a very helpful database that can be used in biology. And the first thing that we need to ask ourselves is, what are GWAS? So GWAS is a genome-wide association studies. They are hypothesis-free methods for identifying association between genetic regions and traits. So it's been long known that genetic variation between individuals can cause difference in phenotypes. A phenotype is essentially the shown trait, what we see as human beings. And these causal variants, and those which are tightly linked to the region of the chromosome, are therefore present at higher frequencies in cases, so individuals with the trait than in controls versus individual without the trait. So essentially that's a lot of jargon that is just saying if you have the genomic trait on a chromosome, it is going to be more adequately expressed in those with the genetic trait than those in the controls that don't have the trait. So variants associated with the disease will be found at a higher frequency in the cases than in the controls, like we just said, and statistical analysis is carried out to indicate how likely that variant is to be associated with said trait. So a p-value, which is just a statistical value of quantifying this research, is the significance of the difference in frequency of the allele tested between the cases and the controls. Essentially, it shows the probability that the allele is to likely be associated with said trait. And a lot of times these results are expressed in a Manhattan plot, which is shown on this slide. It's called a Manhattan plot because the different varying levels can sometimes look like the skyline of Manhattan. So about the GWAS catalog, it was founded by the National Human Genome Research Institute in 2008. It is responsible for the rapid increase of the number of published genome-wide association studies, and these studies provide an unprecedented opportunity to investigate the impact of common variants on complex diseases. So it provides a consistent, searchable, and freely available database of published SNP trait associations. They can be easily integrated with other resources, and they can be accessed by scientists and other people worldwide. It's very accurate, and the amount of information that this specific catalog has on it is exponential. They also publish this iconic diagram with the SNP trait associations mapped on the individual human genome, and so it will give you the specific chromosome location displayed on the human karyotype, which is very cool. So what do we use GWAS for? Since we've established that they're a well-effective method for identifying genetic loci associated with common diseases or traits, GWAS involve the analysis of at least hundreds of thousands of variants across the genome in large groups of individuals. They are often split into cases and controls to identify variants, associations with the trait of interest. The majority of variants identified by GWAS are assumed to be casual, but to tag a link of linkage disequilibrium containing one or more functional variants. Linkage disequilibrium refers to the non-random association of alleles at two or more loci in a general population. When alleles are in linkage disequilibrium, haplotypes do not occur at the expected frequencies. So why do we need the GWAS catalog? Genome-wide association studies are often the first choice approach for investigating links between genotype and phenotype. This is because they are an effective method of identifying associations between the actual chromosomal region where the trait occurs and the actual trait that is expressed in the individual. GWAS have been particularly useful in identifying loci associated with complex diseases where there are multiple different loci that can cause said disease, and they are also able to detect relatively small genetic defects. With this wealth of useful genotype-phenotype mapping data available from numerous studies in the literature, it is important to have a central repository where all of this data is stored and accessible to the scientific community, and that's what the GWAS catalog does. So right now we're going to click on this link and it'll take you to the actual website where the GWAS catalog is, and I'm just going to walk you guys through it really quick. It's pretty simple. So you have your search, your diagram, which is what we talked about earlier, and you actually have the ability to download the entire catalog into whatever format that you want. They have some very useful educational tools for learning how to use the actual database. It's all going to be under the About page. You can go to the Methods page and see that. We can go to different, they have a Frequently Asked Questions page right here. And this will tell you everything about how to access the catalog, how you can use the diagram, how you should cite the catalog. And also, when you learn how to access the GWAS catalog, it will take you to an introductory video. I'm not going to click on that right now, but this will walk you through everything on how to use the entire catalog, which is really exciting. So right here, if we wanted to search something, so we were looking at this semester the SLC 6A1 gene. And so we can type that into the search bar right up here. And so it'll give us the gene right here. So we have two different genes that have the SLC6A1. We're looking specifically at that gene, and it'll also give you a list of 5 different variants right here associated with that gene. And you can click on any of these and it'll bring up all the information about it. There's also an option that can bring up different publications about the gene that you're specifically looking at, and that's really interesting. And the last thing that I wanted to show you guys was the diagram. It might take a minute to download because it is such a big file, because it does map the entire human karyotype, just where all the different traits occur and everything like that. And you can apply different filters down here to look at just specific diseases, like if you wanted to look at just cardiovascular diseases. Right here, there's over 1,000, and you can just line up the specific color trait there to the trait that you're looking for, the SNP, and it'll pull it up. So right here on this part of that chromosome, there is a trait for inflammatory bowel disease, and over here there's total cholesterol measurement. It's very interesting, very useful, and it's laid out in a very concise way. That's the entire diagram right there. There's hundreds of thousands of different things that you can look at, so it's very interesting. And that is the GWAS catalog.